This review is meant to help prepare for the Biology Keystone exam. At this point, we are on topic 7 of our 9 topics, and we are looking at Mendelian genetics and other types of genetics as well. This review should help you answer questions 25, 26, 27, and 32 in your review packet. So to start with a little bit of background on genetics, I just want to mention the name Gregor Mendel. He is the person who we call the father of genetics. Mendel worked in a monastery and he grew pea plants and he tried to cultivate these pea plants, and what he noticed is there are certain pea plant characteristics that he saw over and over again. So he looked at things like the color of the flower. Did a pea plant have purple flowers or white flowers? Where were the flowers positioned on the plant? What was the color of the seeds? What was the texture of the seeds? What was the shape of the pea pod? What was the color of the pea pod? And what was the plant's height? Mendel was able to breed pure varieties of plants, and then what he would actually do is he'd cross-pollinate them. So he'd say, hey, I have this one that's guaranteed to be purple on its own, and I have this one that's guaranteed to be white on its own. I'm going to have them reproduce with each other. I'm going to take some of the pollen from the one and put it on the stigma of the other, and I'm going to see what their babies look like. And he did many trials of this work through many generations. He'd collect the pea seeds, he'd plant them, and he'd count them from there. And what Mendel noticed is that certain traits were passed down in certain ways, and sometimes it appears they were hidden or they might have disappeared. So with genetics, there's a lot of crazy vocab. And I'm going to kind of walk us through with this example down here. So we're going to look at seed color as one of Mendel's traits. We have yellow and we have green. And that's what we're going to be focusing on. As I go through all these terms, I'm going to keep linking them back to this idea. So dominant, if you hear anything about a dominant gene or a dominant trait, this is the one that will be visible. This is the one that shows up. This is the one that does not get masked. In the case of seeds for pea plants, yellow is the dominant trait. Recessive means that it can be hidden. It doesn't mean it's never present. It just means that if there's a dominant and a recessive together, the recessive trait will not show up. In the case of the seed color for pea plants, that would be green. We have a genotype. This is the actual genetic makeup or the alleles of you. So remember, think about DNA. You are really made up of A's, T's, G's, and C's, and that codes for proteins. Your hair is a protein, your eyes are a protein. The main parts of you are proteins. So your genotype is really looking at what makes you up. In genetics, we use a one-letter designation to represent an allele or a gene, something that codes for it. So in this case, I'm using Y because it's the dominant color. Phenotype is related to genotype. This is your physical appearance. Your phenotype exists because of the DNA genotype you have. In the case of our pea seeds, we either have yellow as a physical appearance for the seed color, or we have green as a physical appearance of the seed color. When we describe the genotype of an organism, we can also throw out these two words. We have homozygous. So homo, that prefix means the same. So what happens in the case of a homozygous individual, they have two of the same allele. The reason we have two is because you get half from your mom and half from your dad. So you get one set from mom, one set from dad, so you get two sets. In the case of homozygous here, we could be homozygous for the dominant allele, the capital Y, or we could be homozygous for the recessive allele, the lowercase y. So our genotype could either be homozygous dominant, big Y, or homozygous recessive, little y. Lastly, if you're one of each, you have a dominant allele and a recessive allele, you have a big Y and a little y, we would call you heterozygous, because hetero refers to different. You have two different alleles here. When we are talking about these plants, or peas, or any organism, whether it's cows or horses or whatever the case may be, we have ways of describing the different generations. So we have P for the parental generation. That is the first generation that we're observing. So in this case, continuing right along with seed color, we have pure breeding yellow. So this would be homozygous dominant, big Y. We're going to cross that or mate that with a plant that would normally produce green seeds, pure breeding green line. So that would be homozygous recessive, little y. The babies of that parental generation, we call it the F1 generation. And F1 really stands for first filial. Filia, filius, referring to Latin Greek for son. And we can see here, in this case, all the babies turn out to be yellow. We'll talk about how that happens. And then if you continue it down further, we would call that the F2 generation, second filial. And you can see here, I have some yellow and some green. So the way that we're able to actually figure out why do they all end up being yellow in the F1, why are they yellow and the green in the F2, or questions like, why can't you have brown eyes if both your parents have blue eyes? Those genetics questions, we like to look at Punnett squares to help us figure them out. And I'm sure in your biology class you remember doing these Punnett squares to help you with your practice problems. So basically, what you're doing is you're taking your parents' DNA, or the P generation here, and you're trying to cut their DNA in half. And the reason they want to cut their DNA in half is because you're really making sperm and egg. And sperm and egg are a product of meiosis, and as we talked about in previous podcasts, we want to make sure that we have half the DNA so that our offspring have the correct diploid number. So what we do is we take those possible half DNA combinations and we put them on the outside of a box for the parents. Female can be up top, male can be on the side, or vice versa. It doesn't really matter. And then what you do is you cross those outside combinations, and then inside the box you figure out what the babies could be. One important thing is this is just a probability of what is likely to happen or what is not likely to happen. The way I like to think about it is a coin flip. The chance of having a boy or a girl is 50-50. So if you have two children, statistically, one of them would be a boy and one of them a girl. However, you could absolutely have all girls in your family. It's what's probable. It doesn't mean it's definitely going to happen. So just to look at some Punnett square problems, I have some on snakes, and snakes come in all different colors. These are looking at corn snakes, so when I just show you these different words, that's what they actually look like. The simplest type of cross that is ever done is called a monohybrid cross. And the reason it's called monohybrid is you are only looking at one specific trait. In this case, we are crossing a heterozygous individual with another heterozygous individual. You don't have to always cross two heterozygotes, it just helps to see differences. So again, if this is my parent, I split it into the possible combinations along the top, and my second parent, I split it into the possible combinations along the side. What I then do is I fill in the box of the offspring. So this offspring is homozygous dominant, they will show the dominant trait. This offspring is heterozygous. This offspring is heterozygous, and this offspring here is homozygous recessive. So from a phenotypic ratio, what they would look like for my corn snakes, 3 of them are going to be the normal coloration right here, and 1 of them ends up being an albino. For your genotype ratio, that would be the actual genes, the letters that make it up, I get a 1:2:1 ratio. 1 is still normally colored, it's homozygous dominant. 2 are heterozygous, normal coloration, and 1 is homozygous recessive, albino coloration. If I wanted to make things more complicated, I could look at a dihybrid cross, and a dihybrid cross would look at 2 genes or 2 traits. So we're looking at albino versus non-albino, and we're looking at pigment versus kind of no pigment or making pigment. So what you can see here is if I cross 2 heterozygous individuals, they're heterozygous for the A trait, big A, little a, and they're heterozygous for the little trait, big Y, little y, I get even more combinations for those genes. Because I could have big A, big Y, but I could also have big A, little y. I could have little a, big Y, and I could have little a, little y. So I'm looking at whatever combinations of those alleles I could get. And since this one is the exact same, we throw those on the side. If I go through here, these are all of the possibilities of the offspring. So instead of having 4, there are 16 different combinations. If I look at the phenotypes, what they physically look like, 9 of these snakes are going to have the normal coloration. 3 of the snakes are going to have the anary coloration. 3 of the snakes are going to be a regular albino. And 1 snake is going to be the snowy albino. So that's my phenotypic ratio. My genotypic ratio is a little more complicated. You can see it's a 1:2:1:2:4:2:1:2:1. But basically what we're looking at here is in a dihybrid cross, you're looking at this magic phenotypic ratio, 9:3 to 3:1, if you cross two heterozygous individuals, and for a monohybrid cross, you get a 3:1 ratio if you cross two heterozygous individuals. So out of all of Mendel's trials and all of his ideas and his experiments and over, he came up with some really important ideas. The first one is the principle of dominance, or the idea about dominance, that there are certain alleles that are just dominant. Dominant does not mean better. Dominant does not mean more prevalent in the population. Dominant just means that if dominant and recessive are both present in your body, you are only going to show dominant. Tongue rolling. It is not better to be a tongue roller than a non-tongue roller. There are not necessarily in a given population more people who can roll their tongues than can't roll their tongues. However, being able to roll your tongue, and I'm sure you're all trying this right now, that is a dominant trait. So if you have a gene for tongue rolling and you have a gene that doesn't let you roll your tongue in your cells, the gene for tongue rolling is dominant. That's what shows up. If you can't roll your tongue, then you would have two copies of the recessive gene. Mendel also talked about the idea of segregation. Segregation is similar to separation. What this means is during meiosis, chromosomes are going to separate so that each parent only donates half of the genetic material. You want two haploid cells to come together to turn into a regular diploid cell, which will make that individual up. There's also the idea of independent assortment. And what that basically says is that not all of your mom's chromosomes, split perfectly. It's a completely random process. And that's why you don't look exactly the same as all of your siblings. Every time your father produces sperm, it's completely random which of his genes get thrown into each sperm cell. Every time your mom releases an egg, it's completely random as to which genes go into that egg cell. So if you look at this picture here, they're showing you the two parents, independent assortment. In this case, big A ended up on the left and little a ended up on the right. It could have flip-flopped. And then what we see is it's those random joinings. You can see here, this sperm goes to these two children, this sperm goes to those two children. This egg goes to those two children, this egg could go to those two children. So it's showing you these are all of the potential combinations. And that's why you don't look exactly like your siblings, because of segregation and independent assortment. Now, when Mendel did his work, he did it way before Darwin, way before evolution, way before molecular biology took off. So during Mendel's time, he only really had simple dominance. What we've seen since then is there are more complex patterns that explain genetics. One of them is called incomplete dominance. So I said before, dominant always masks a recessive allele. Not really the case. In incomplete dominance, it doesn't perfectly cover it up. So I have two examples here, a 4 o'clock flower. Normally, red would be dominant and white would be recessive. So if we were thinking about basic genetics, Mendel's genetics, well, if red's dominant and I bred them, then the baby should be red. However, in this case, it's a blending of the two traits. So red and white makes a pink flower. You can see here, if I breed these two pink flowers together, so this is one pink flower, this is another pink flower, what I see is that red reseparates and that white reseparates. I have to have two copies of the dominant allele to be purely red. I have to have two copies of the recessive allele to be purely white, and I need to be heterozygous. I need a dominant copy and a recessive copy to be the blended incomplete trait. In human beings, hair type would be an example. If you have curly hair, homozygous dominant. If you have straight hair, you're homozygous recessive. If a person with curly hair marries and mates with someone with straight hair, then their child will be heterozygous and they will have wavy hair. There's also codominance. Think of together. In codominance, both alleles are dominant and both alleles show up evenly. So in human beings, we have the blood groups. We have A, B, AB, and O. If you have AB blood type, which is here and here, you have a copy of the A allele and you have a copy of the B allele. A is not dominant to B, nor is B dominant to A, so they both show up together. However, there's a third allele, there's I here, and A is dominant to I, and B is dominant to I. So you can see here, if this person has IA little I, their blood type is A. You have to have both copies here to be O. But again, AB codominant. Rowan animals are another example. If I have a dark, kind of reddish colored and a white, what ends up happening with rowan animals is they have white and brown together. It's like a speckled kind of appearance or a pattern appearance. That's an example of codominance. So that's what they're showing you here. I have one copy of this allele, I have one copy of that allele. One is not dominant to the other, they both show up equally. If we look at human beings, we can look at gender. And the reason I said before it's a 50-50 shot of having a boy or girl deals not with mom, deals with dad. So we talked about chromosomes, and chromosomes are really, really big pieces of DNA. And the human diploid number for our chromosomes is 46. You have 22 sets of autosomes, and then you have one extra set. Your 23rd set is the set of chromosomes that determine your gender. If you have two X's, you are a girl. If you have an X and a Y, you are a boy. So for males, you're the ones that determine the gender of your baby. If you donate an X, it goes with mom's X to make a girl. If you donate a Y, it goes with mom's X to make a boy. What we've learned is there are certain traits or certain genes that are on the X chromosome, and having a defective copy of them leads to disorders. Color blindness or color deficiency is an example. More men tend to have color blindness than women. And the reason they do is because women have two X's. So even if one of their X's is crappy, they still have another chance to have a good X to cover it up. Men only get one X. They have an X and a Y chromosome. So if their X is defective, they have to show it. They aren't going to have a dominant allele to mask it. Hemophilia is another example. Severe combined immunodeficiency example, and there are plenty others. So if you look here, instead of dealing with like an A and an A or a B and a B, we are using X and we are using Y. X denotes the X chromosome and Y denotes the Y chromosome. You can see here having this little h is showing you that they are carrying the recessive gene for hemophilia. But you can see here, this woman, big X with a little h. So she does have the gene for hemophilia, but she does not actually express or show the disease because she has a good copy of the gene. If a male over here is X little hy, he has hemophilia. So what we're just looking at here is just another way to look at it, just putting a different spin on this whole genetics issue. For human chromosomes, we can check out if there are any problems on a larger level by doing something called a karyotype. And a karyotype is where they actually just sort out all the chromosomes of one of your cells, and then they kind of take a picture of them and see what's going on. You can see the XY here is showing you that it's a male. Down here, I have my sets of chromosomes, blah, 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 blah. If you see here, I have two X's, no Y. That's a regular female. If you see here, everything looks hunky-dory. I have 2, 2, 2, 2, 2. But here, on my 21st chromosome, I have not 1, not 2, but 3. So that is actually a genetic disease called trisomy 21. And then down here, you can see I have everything's normal. I have two X's and a Y. Now either you have two X's and an X or a Y, you don't normally have both. That's another disease called Klinefelter syndrome. And you can see over here, something is very, very wrong. I have 3 of each chromosome. A whole extra set of chromosomes that doesn't have a disease name associated with it, because in all honesty, that is completely fatal for human beings. It would never, ever work. So, non-disjunction, we talked about that before. Non-disjunction is the idea of chromosomes failing to separate properly during mitosis or meiosis. In the case of looking at making an offspring, we're focusing on meiosis. So it could happen either in meiosis 1 or it could happen down here in meiosis 2. If you have an extra chromosome or you're missing a chromosome, we have a couple of different diseases, and there are many more, but I just pulled some more common ones. You have Down syndrome, you have Klinefelter syndrome, and you have Turner syndrome. If you have an extra set or a missing set of chromosomes, again, that's pretty much fatal to human beings, fatal to most animals. They wouldn't be able to survive. In terms of genetic engineering, basically the idea is to take a gene of interest, put it in something else, and have them make it for us. It's altering an organism's genome. It's altering an organism's DNA. However, due to some of the things that we don't really understand about it, there's a lot of controversy and there's a lot of fear surrounding whether or not this is something we should be doing. But an overview, really simply, how is it done? There's a gene of interest, something we like. I always go with my maple pig example. I love maple syrup on bacon and I love maple syrup on sausage in the morning. So my dream would be to take a gene I like, the gene for maple syrup that makes that delicious sugar. Tweak it if I need to modify it to make it pig friendly, and then I carefully put it inside of a pig. I'd have to put it inside baby pig cells, a tiny pig embryo, and then hopefully the idea would be that the pig would see that piece of DNA, would see that piece from maple syrup and make maple syrup right next to its proteins, and then I could get a pig that tastes just like maple syrup. There's a lot of good that comes out of genetic engineering. The idea we can get bigger produce or more tasty produce. We can make medicines. So insulin for diabetics used to be slaughtering animals and taking the insulin from them. Costly, timely, not the best thing. What we can do now is we can take the gene for human insulin, stick it in a bacteria, have that bacteria make our insulin much better, much cheaper. We can make food more resistant to cold, to drought, to disease, to other things. We can make it more colorful. So there's a lot of benefits there. However, there is some negatives associated with genetic engineering. We don't always know what we're adding into food. A lot of it could be, hey, there's a protein from something like peanuts. A lot of people are allergic to nuts. If they take a gene from a peanut, that could also produce the antigens that would cause someone to go into an allergic response. So that would be bad. FDA really doesn't label nearly as much as it needs to. And that's an issue because, again, you don't know if the food that you're eating is genetically modified. Chances are it probably is. And you don't know what they've tweaked. Did they put another organism's DNA in it that you might have some sort of response to? There's also the idea of building up resistance to chemicals. So we create a farm plant, a crop, and this crop is resistant to a certain type of insecticide, so we can spray everything else. Well, if that plant perhaps is able to breed with one of the weeds that you don't like, it can cross-pollinate. Now your weed is also resistant to that insecticide. And there's also the idea of unknown ecosystem reactions. We don't really understand exactly how something might affect the ecosystem. If we tweak the grass to be disease resistant, maybe the grasshopper won't eat it. And if the grasshopper no longer eats it because it doesn't find it tasty, then the snake loses its food source and so on. So that could be a very negative thing there. So, to sum everything up, we have the idea of genetics, and genetics explain how traits get passed down from generation to generation. How come blonde hair runs in your family? How come most of the people in your family are on the shorter side? But it also explains genetic variation. Why don't you look exactly like your sister, or why don't you look exactly like your father? Mendel has some important ideas of dominance. Certain traits are dominant over others. Segregation, chromosomes separate, and independent assortment. This gene goes here, that gene goes there, to help explain why genetics works the way it does. However, since Mendel's time, we've learned that it's not a clear-cut system. And human beings, like everything else, are affected by genetics, and some of our diseases or disorders are linked to problems in our genetic makeup. And we can use genetic engineering to modify organisms, but we need to be mindful of some of the things we don't understand about it.